Oh, now it's not. Hi, I'm Miss Sloan, and this is Honors Biology. And right now, we are going to talk about how large I am in the middle of the screen. Right now, we're going to be talking about chapter 27, but it's the second half of it. And it's all about evolution. I didn't make a list, so let's make a running list as we go along. Okay. Um, the first thing we did is we kind of backed up and did a little review, and I want to do that with you now because that's part of your final, which is starting tomorrow. So if we look at the steps of life, we want to remember that the early Earth, right, was what kind of environment? Reducing. Good. What gas was missing? Yeah, no free oxygen. So no O2, but you had like oxygen and H2O and water, carbon monoxide, but there was no free oxygen like what you and I breathe. And as a result of that, it was conducive to what? Building, not breaking, right? And those early gases could have come out of and there was rain, and we had ocean, and we had energy, things like UV radiation and radiation and molten magma. And with that energy, you were to take inorganic molecules and make organic molecules. And there was a man who did an experiment, and it didn't take him a billion years. It just took him less than 2 weeks. And he was able to make inorganic into organic through his apparatus where he cycled those gases around with energy. What was his name? Miller. Good. And then once you have the building blocks, then you could have the same energy that we talked about, okay, could also help you to build the organic polymers. So the 4 important organic molecules that we talked about, let's try to do them in the order we talked about them. We talked about carbohydrates, and what are carbohydrates built out of? Sugars, right? Carbohydrates are built out of sugars. And I'm kind of backing up here just because we have a final. So carbohydrates are built out of sugars. What's the name of the reaction that joins them together? Dehydration, synthesis, and breaks them apart. And that is the same for all 4 important organic molecules, right? Okay, so carbohydrates. Then we talked about lipids. And there were 2 categories of lipids. What were the 2 categories? Fats and steroids. Steroids are made out of what? 4 rings. And then fats, you have 1 and 3 somethings. What's the 1? Glycerol. And 3 fatty acid chains. Those fatty acid chains could be or saturated or unsaturated. Where do we tend to find saturated fats? From animals. And at room temperature, is it raining? And at room temperature, it's a solid. Whereas unsaturated fats we usually find from, they are liquids, and you get them from plants. Good. Okay, so okay, proteins. What are proteins built out of? Amino acids. And this is an amino acid right here. Remember the amino group, central carbon, HR, and the carboxyl group. Does that look familiar? And you remember the special bond when you join amino acids together? It's called a peptide bond. Perfect. Okay. And then the 4th important organic molecule is what was that? We did carbs. You're saying it. Carbs. We did lipids. And now we've talked about. Oh, okay, great. Thank you. Proteins. And then what was our last one? Was nucleic acids. And nucleic acids are built out of nucleotides. Perfect. And nucleotides are built out of a base, phosphate group, and sugar. Yeah, nitrogenous base, sugar, and a phosphate group. Do you remember what the 2 sugars were? Ribose and deoxyribose. And that's where we remember RNA and DNA. There you go. Just a little bit of review on that. So we have our polymers. Then we use our polymers to build our cell. Prokaryotic cells are more simple, right? Eukaryotic cells have a lot of compartmentalization. Who has a larger surface area to volume ratio? Prokaryotes, because they're smaller. Good. And was the first cell an autotroph or a heterotroph? Heterotroph. Good. And then you got eukaryotic cells. Do you remember the endosymbiotic hypothesis? How one prokaryote went to live in another prokaryote? Does that sound familiar? Okay, and evidence of that is mitochondria and chloroplast. We've talked about this, right? Or is all my biology coming together? That they have their own DNA and they're self-replicating. The DNA is circular like prokaryotes. Does that all sound familiar? Perfect. Okay, So those were the steps of life. And then when we started in this whole unit, we kind of talked about both microevolution and macroevolution. And microevolution, let's see if I can go up here. Microevolution is change within the species, right? And macroevolution is change to a whole new species. Perfect. And we revisited that there in the last bit of our chapter. But what we went into next was the evidence for this evolution over time. So we had different pieces of evidence that we discussed. See if I can make it go there. Go there. Go there. Is it there? No, there it is. Okay. So we talked about fossils, biogeographical evidence, biochemical, anatomical, and I still included a biology one, but we talked about the geologic time scale, yes? So you can cross that out, sorry. I always want to put in this picture instead of the other one. So on fossils, let me come back to fossils, just put a pin in that for right now. Let's talk about this one, biogeographical. Think about what is some of the evidence we have for biogeographical evidence. Pangea, good. Remember how all the continents fit together, and we could see continuity of fossils within that. We could see like Antarctica has fossils of amphibians, so we know that Antarctica wasn't always in a cold location, right? So that's your biogeographical. Anatomical, there are 3 pieces of anatomical evidence. See if you can remember the 3 pieces of anatomical. Homologous structures, vestigial structures, embryo. Yeah, analogous is, remember we said, not a piece of evidence, right? So homologous structures is what you're seeing in this picture right here. Having that same bone structure where you have humerus, radius ulna, carpals, metacarpals, phalanges. If our bone structure is similar, maybe some of our phalanges are longer, maybe some of the humerus, they're shorter, but you can still see that type of bone structure, then probably we came from a common ancestor, and that you had slight adaptations for whatever environment you were living in. So that would be homologous structure. Homo means same, right? So same anatomy. If we have the same kind of bones, then we must have similar DNA too that codes for that. We don't talk about analogous structures as evidence because analogous structure is just function, right? So I walk on legs and spiders walk on legs, but do we have a common ancestor? No, we just use legs to walk, right? Dolphins swim in the water and they have fins, and tuna fish, they have fins, but is their fin structure the same? No, they just use it because they're swimming, right? So we want to make sure we don't use, if I ask you that question, don't use analogous structures as evidence. All you're saying is I'm living in my environment the same way. It's when the anatomy is the same, that's pointing to a common ancestor. Okay, then this right here is from a whale. A whale is a good example of a vestigial structure. It has something that's vestigial. What is that? Tell me. Yeah, it has a hip bone, part of a femur. So what does that tell you that a whale used to do? Used to walk on land, right? So that points to its ancestral past. That shows us there was change over time. Salamanders that have withered eyes, it's like, were they born with withered eyes? I mean, you could be, but more than likely that salamander or that species of salamander lived where they needed eyes. But then if they migrated into a cave, eyes don't become as important. And so their eyes don't keep them alive or increase their fitness. They don't pass on their genes even more, so they become withered. That tells you where they could have come from. That is a vestigial structure. And then the third one is just comparing embryos. The more similar the embryos look, probably the more common the ancestor. And then I talked to you about when you're an embryo and developing, it's kind of like evolution on fast forward. What are some of the structures you have when you're an embryo? You have gills and a tail. Yeah. So it's almost like we're going fast on that. So those are all related to anatomy, homologous structures, vestigial structures, and embryological structures. And that's why I have development right here, but it also relates to the genes that code for those things. Okay? Biochemical evidence, this is the one that's used the most right now. When you did your phylogeny, where you did your cladogram, you're either comparing sequences of DNA or sequences in amino acids and proteins, right? And looking to see, hey, if our DNA or our proteins are more similar, then we're more closely related. Okay, so now I want to travel back to fossils. And remember, we said to be a fossil, you have to be how old? Yeah, 10,000 years old, and you have to be something that could get fossilized and found by a human to be called a fossil, right? And we talked about transitional fossils that resemble two different species. And later, the last thing in this unit, we talked about pace of speciation. Remember the two paces of speciation? What was one of them? Punctuated equilibrium, and the other one was phyletic gradualism. And where would you see more of these transitional fossils? Yeah, with gradualism. It's kind of like you're leaving a trail of your evolution, evolving very slowly, whereas punctuated equilibrium, you're jumping around to your new species. And by jumping, we mean like a short time period, like a million years. Yes?
Are we going to on the puzzle those other
directional? Yes, I can go over those if you want to. Okay, and then here, okay, when we talked about fossils, we talked about two main types of dating. So relative dating and absolute dating. Relative dating, you look at what kind of rock layers to do relative dating. Sedimentary rock, and you're just assuming that the layers on top are newer and the layers on the bottom are. And you have to watch out for what kind of zones, though. Subduction zones, right? To make sure that that didn't happen. So that is like relative dating, one to the other. Absolute dating, you're looking at the breakdown of isotopes because we know the speed at which different isotopes break down. So we measure how much of it has broken down relative to what you would find now. And then we can give an actual date for how old something is. We can also use things like index fossils, like this is the trilobite, and it's only found in the Paleozoic era. So we can use that to help identify a certain time period. And then speaking of time periods, let's go over those time periods. Okay. So the era you are living in right now, it is the center of your world, is called the what era? Cenozoic. And it's this moment back how many millions of years? 65 million years ago. And we're going to remember that's when we drive our car, right? Then what's the era right before the Cenozoic? It's in the Mesozoic in the middle, okay? So the Mesozoic era, it ended 65 million years ago when the era we're in right now is the Cenozoic era, but it started about how long ago? Do you remember? Yeah, 248. So you're in the 200s of millions, yes? And that Mesozoic era in the middle is when you had what? Dinosaurs. Good. What was the era before the Mesozoic era? Paleozoic era. So and it has the years right here. Okay. The Paleozoic era is about what? When did it start? 542 million years. So the way I remember that is dinner time, school's out, and speed limit. Okay. And as long as you have those. Then everything before the Paleozoic era is called what? Precambrian. And that's because the very first period, it's not on here, but the very first period in the Paleozoic era is the Cambrian. So keep in mind that Precambrian is where there is most time. Do you remember how I gave you that 24-hour clock for perspective? It's like almost all the time, right, of our 24-hour clock. And in the Paleozoic era, that's when we first had the Earth. How old's the Earth? Yeah, and you had first life. How old is the first fossil? Yeah, about 3.6 billion. So that's that whole evolution of life thing that we talked about at the very beginning, the stair steps, right? And then you're going all the way up to 542 million years. And basically, at this point, you have evolved to the point of invertebrates. So that means without a what? Without a backbone, and plants that are just algae. You're at algae to invertebrates. But here, you're going to go plant-wise, you're going to go in the Paleozoic era, you're going to go from those algae all the way up to Christmas trees or Hanukkah bushes, which we call what? Starts with a G. Remember? Gymnosperms. Gymnosperms, okay? And then you're going all the way from invertebrates all the way to early what? Reptiles. Okay? So that's a big leap in the Paleozoic era. Huge leap in the Paleozoic era. Algae to gymnosperms, all the way to trees. Algae to trees, and no backbone all the way to the beginning of reptiles. Yes?
Are we going to know like every transition, or just that it goes from early mammals to like herbs and the Cenozoic?
Well, I'm going through the ones you would know right now. Okay? Then in your Mesozoic era here in the middle, you have early reptiles to early, what are we? Mammals, animal wise. Okay? And then you're going from gymnosperms to flowering plants. It starts with an A. Do you remember what that's called? Angiosperms. Bringing some flowers to Angie. Angiosperms. Bringing some flowers to Angie. Okay? Angiosperms. Okay? And then in the Cenozoic era, you're going from angiosperms, flowering plants, to herbs, and you're going from early mammals to us, to humans. Okay, and I think if you know your bookmarks, you know you ought to be okay. If you know you are an invertebrate in the Paleozoic era, then probably you're going to get some fish in there, right? And some amphibians before your reptiles. So we kind of said invertebrates, fish, amphibians, reptiles. That's all your Paleozoic era. Early reptiles all the way to early mammals, right? Then early mammals to humans. Remember how I kind of broke it down for you like that? And then here you're going from nothing, no living thing, right? And we talked about you end at algae, algae to gymnosperms, gymnosperms to angiosperms, and angiosperms all the way up to herbs. Okay. So then that was part of and including the geologic time scale, and it relates to fossils as well. Then we took a day where we just talked especially about biochemical evidence, the sequence of amino acids or the sequence of DNA, and we talked about building cladograms. So your POGL was kind of hard about like rearranging and getting it. There's nothing I'm going to give you on a test that it's that hard. Okay, so don't stress out about that. But remember, the gist of cladograms is like, are you still into club, right? Or do you get dropped out? So it's like from here on up, not including the fish, everybody has at least what? Lungs. So we're going to assume that all of these organisms evolved after fish, right? Because they all have what the fish has, and then they have one more thing. They have lungs, right? But then we dropped off claws and nails because who doesn't have that? You know what that is? Like a salamander. Yeah. Okay. And then from here on up, everybody has hair. So we just keep getting, it's like we have all of these things. Both us and a gorilla has all of these things, right? But the one big difference here, according to this chart, is just that we're upright and we're walking. Okay, so cladograms, you can do that same thing, just looking at DNA and saying, okay, this much DNA is the same. Okay, this group has this gene or this protein. And then we keep dropping them off if they don't have that. Okay, so that is kind of setting up the big picture about evolution and change over time. But then we said, okay, how do you actually measure that? And that's when we started talking about population genetics. And we realized that an individual does not evolve. An individual does not evolve, but a population does. And so we looked at the five, right? The five things that can contribute to change in allele frequency. Now remember, how many alleles do we have for each of our traits? 2. And we haven't gotten into the genetics yet, but you know you got one from mom and one from dad. And remember how in the class, I am sure in each one of my classes, we counted how many people were in the class and we figured out how many of you had blue eyes in the class. Remember that? And we know the blue-eyed people had to have the recessive trait. And we said, okay, if another class came in, would it change the percentage of blue-eyed people if another class came into this class right now? Yes, it would. So population-wise, whenever your allele frequency changes, evolution occurs. So we talked about the 5 things that can contribute to evolution. So let's go through and make sure we understand them. And because there's a couple significant things on it. The first one, starting with the pinky, what is this one? Small population. And the reason why small populations are more likely to contribute to evolution is because they're more susceptible to chance events. And that's called genetic drift. Genetic drift. And we talked about two in particular. Do you remember the two types of genetic drift we talked about? Remember? Bottleneck and founder's effect. A bottleneck is if you have some crisis, right, that you go through. Let's say a fire comes through a community, but if it's a very small community, you can wipe out a lot of alleles permanently through that crisis, where if you have a larger community, you're probably going to save some of those alleles, right? And then founder's effect is when you start a new population. Look around in this room. Do we represent Oak Park High School if we were a founding committee of new people on a new place? Not necessarily in the right percentages, right? And we saw that with the Amish people in Pennsylvania. Remember, they had a 5-fingered or 6-fingered dwarf. So they have a higher incident of 6-fingered dwarfism because they founded the Amish community in Pennsylvania than you would in the regular population. Okay, so small populations definitely change allele frequencies. Non-random mating was the ring finger, right? And that means sexual selection. I am choosing you for these traits. It's not what kind of mating. Random red cup mating, where any vagina or penis will do. Oh, it's on YouTube. Any reproductive structure will do. You're being very specific about which one you're going to mate with. That will change allele frequencies. Mutations change DNA, so that will change frequencies. Gene flow. What is gene flow? Immigration and emigration, right? So if you have new blood come in, that could affect your allele frequencies. If you have people move away, that could change your allele frequency. Natural selection, that thumb was special because this is the only one that leads to what? Adaptations. Those are those traits that make one organism better suited to that environment than another. Right? To be a giraffe, what is an adaptation that's good for a giraffe? A long neck. To be a tiger, what's an adaptation for a tiger? Sharp teeth, they're fast, they can hide, right? All of those are adaptations. Who is selecting who's best fit for the environment? Nature, natural selection. And who came up with that idea? Starts with a D. Darwin. Okay. When we measured that, we used two equations. Okay. So allele frequencies and genotypic frequencies. So allele frequencies would be like how many blue-eyed alleles in here versus how many brown-eyed alleles in here, right? And then we could talk about homozygous, heterozygous, or homozygous recessive. So let's just practice one right now just to make sure we feel comfortable with it. There are 2, 4, 6, 8, 10, 2, 4, 6. There's 16 of us in this room. Are you okay with that? 16 in the room. That is our population size. Okay, is 16. Oh, I wonder if I can write on here. Oh, I can. Okay, good. That's our population size. We each are carrying 2 alleles. Do you agree with that? How many of the 16 have blue eyes? I'm standing up because I have blue eyes. Blue-eyed people, stand up. That's it, just the 3 of us? Are we good? Okay, so 3 out of 16 have blue eyes. What are we in this equation? The 3 out of 16 with the blue eyes. Yeah, we are this. We are homozygous recessive because the only way to have blue eyes is to have 2 little b's. Do you agree with that? So what we could say is, okay, I can solve for this right here, q squared. Q squared equals 3 out of 16. So let's find out what 3 out of 16 is. Do you have a calculator with you? So I did 3 divided by 16. Okay, so now what did you guys get? Pardon? Point what? Yeah, that's what I got too. I got 1875. Is that what you got? So now how do we solve for Q? Just take the square root. So now take the square root of that. Can you take the square root of 0.1875? Do y'all have calculators? Did you get it? What'd you get? Okay, so he took the square root and he got Q to equal what? 0.33, is that right? Okay, so now we know if Q equals 0.43, then what does P have to equal? Because P plus Q equals 1. 0.57, I agree with you. So in this little population, in this room right now, that if we were to take both of our alleles that each one of us carry and throw them in the gene pool, there would be like a 57% chance I'd pull out a dominant allele and a what? 43% chance I would pull out a recessive allele. So if I wanted to predict how many in this room were homozygous dominant for brown eyes, then I would just have to do p squared, right? So it would just be 0.57 times 0.57. That would give me p squared times 16 people in the room. And 2 times p times q times 16 people in the room would tell me how many heterozygotes were in the room. Why do we care about that? Because if this allele frequency changes, if for some reason Q drops down to 0.3, that means P has to be what? 0.7. If the allele frequencies change, what is occurring? What could cause allele frequencies to change? Small population, non-random mating, mutations, gene flow, natural selection. Okay, that's all evolution is. Does that make sense? Okay, perfect. Can I erase? Can I? Okay. Can I erase? Yes, I can. Yes.
For the answers, if we were plugging into that equation, would you always put it in about percentage, or would you like asking the question like what is like?
Usually P and Q are given as a decimal. Yeah, as a decimal. You can write it either way. You can write 73% or 0.73. Either way, I'll totally know what it means. All right, and then, so remember, assumptions of Hardy-Weinberg says no selection, that would be the thumb, no mutation, that would be the middle finger, no migration, that would be the pointer finger, always a large population, that's right, the pinky, and random mating, ring finger, in order to keep the allele frequency change, or allele frequencies to freeze. And if you can't freeze it, and because it's so difficult, then evolution is occurring. Okay, and then go? Yes, it is. It's hard to meet those 5 conditions. So then we talked a little bit about adaptations again that make them better suited. So you have selection here because those seals who had more blubber, then they stayed alive because they weren't freezing, right? And so that increased their fitness. All right, and then that brought us to like the types of speciation. Allopatric speciation always starts with a what? Barrier. So you get separated, you have different initial gene frequencies, you have different mutations, different selection pressures on either side. And the example we use for allopatric speciation, besides the dewlaps, when we looked at that, the anole lizards, but we looked at salamanders migrating from Northern California to Southern California, and they kept accruing changes. So even though the Central Valley wasn't a barrier to them anymore, by the time they got to the bottom, they had accrued so many differences that they were reproductively isolated and they were their own species. We looked at some examples about finches and going to other islands where there was less competition and maybe acquiring some adaptations there. So if they return, it decreased competition. And that is one form. That would be allopatric speciation because they had a barrier between them. And then we said you could have sympatric speciation where you were side by side, yet you changed. And usually that's due to what? When you're side by side. Changes in chromosomes, exactly. It could have to do with diet, like, you've changed a little bit what you're eating. I've decided to eat newer leaves instead of the older leaves that everybody else is eating because there's less what? Competition. And so if over time beak changes in my family line, then I can accrue and be reproductively isolated. And I gave you a little diagram to discuss where we looked at when you see that barrier, that's allopatric. When it's within side by side, that's sympatric. Another word I want to get in your head is adaptive radiation. Remember where it just like a whole bunch of variety exploiting all the different niches. And then the last thing you already had, we already went over, and that was the pace of it. Okay, good job. Anybody have any questions? Yes.
There's 3 different Raphael and Vogel, the different types.
Oh, yeah. Directional selection is favoring one extreme, and that can lead to speciation. You can come on in. Disruptive is 2 extremes, and that can also lead to speciation. And stabilizing is the norm. It does not lead to speciation. Directional, disruptive, and stabilizing. Yep, those are types of selection. Okay, I'm going to go ahead and